Alright guys, so this next lesson is going to cover 3 different types of genetics problems. We're going to look at dihybrid, which dihybrid is looking at 2 traits at the same time. And we're going to look at something that you have not done before in biology. We're going to look at what I just call the other problems. And so if you go back to your packet, you have your note packet that has Punnett squares at the top, and it's got some of the same information pretty much down at the bottom. It talks about the dihybrid being 2 traits at a time. And then if you flip over to the back of that front page, you're going to have just a little bit of information on the other types of problems. Okay, so we could technically look at 3 traits, 4 traits, 5 traits. We can look at multiple traits, but we're going to look at a shortcut. We're also going to look at some problems that kind of have us thinking backwards. And this is why it's really important to understand genetics concepts, but also understand Mendelian ratios, and your life is going to be a lot easier. Okay. Now the dihybrid should not be new to you. This is something we did do freshman year. It's pretty much just a slight expansion over what we did in the last set of problems. So like before, we're going to talk about how to do them. We're going to do some examples in your note packet, and then you're also going to have that Dragon Genetics packet, and there's some more problems that you can do in there. So when we take a quiz, our next quiz, these are the types of problems that you will see. Alright, so we're going to start with the dihybrids, and so if you turn in your note packet, we're going to pick up where we left off last time. After number 6, there's a little section about dihybrids. And so we're going to look at number 7 and number 8, just to kind of do some practice. So I do want to remind you that the same basic 5 steps still apply here. I think the key is especially important in the dihybrid. I think it's even more important here. We still have to figure out what the parent genotypes are, but because we're looking at 2 traits, a parent genotype is going to have 4 letters now. It's going to have 2 for trait number 1 and 2 for trait number 2. Now step 3, we're still going to separate, because like before, the parents can only give half of their information to the babies, but we are going to add a step. Okay, this is where we're going to add the foiling. You might remember that from freshman year. It's also a concept that you learn in math. So we're going to take a look at that. This is really the only new thing, is we're adding a step when we separate. We can't just split the letters over the box like we did before. We're going to do our cross, and that's where we figure out the offspring genotypes, and then we're going to answer whatever the question is asking us. Okay. Now, just as a quick reminder, because this is sometimes causes confusion with dihybrids, so remember that outside of the Punnett square, this time we're going to have a 16 box. Because if we go from 1 trait to 2 traits, we go from 4 possibilities to 16 possibilities. The outside, what you're putting out there are the possible gamete combinations. What could be in mom's eggs and what could be in dad's sperm. So I just want to remind you that the outside, these are not genotypes. These are possible gamete combinations. And so there's a reason we're going to write them the way that we do. Now, inside represents the offspring genotypes. And like the parents, the genotypes are going to have 4 letters. And we want to put like letters together. So if I'm using the letters A and B, I want to put my A's together, capital A first, and I want to put my B's together, capital B's first. Okay? So sometimes students will just write it, like A, B, and that can get really, really confusing. So you'll see what I'm talking about as we do examples. If you remember how to do these, awesome, but if not, we're going to kind of start from scratch and treat it like you've never done one before. Alright, so as I mentioned, there's going to be 1 extra step involved in the 5 steps, and that is foiling. So the first thing I want us to practice doing is using the FOIL method. So once again, if we're looking at 2 traits, then parents are going to have 4 letters in their genotype. And so if you look at number 7, you have a couple examples. You have an A, B, and C there. We're going to take a look at maybe just A and B. So let's say we have the parent genotype, capital A, lowercase a, capital B, lowercase b. Okay. Now the foiling process, like I said, you've probably seen this in math. I learned it as first, outer, inner, last, You might know it as first, outside, inside, last. It's the same thing either way. But what we cannot do in a dihybrid is we cannot just take these letters and just write capital A, lowercase a, capital B, lowercase b. That is wrong, 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 okay? And if you do that, you're not going to be able to get the question right, okay? You're going to have to understand that if the parents have 4 letters, they're going to be giving their babies 2, okay? Remember that we're going to give half of our genetic information to our offspring. So if mom has 2 A's, she's giving her baby 1 A, and if she has 2 B's, she's giving her baby 1 B. So what we need are all the possible combinations of A's and B's based on that genotype. And so just as a foolproof method, okay, once again, consistency is key. If you always are consistent and you do it the same way, you're less likely to make mistakes. So what you do is you take the first of each letter. And I've been foiling for years and I still draw the arrow, okay? You take the first of each letter, so that baby could get a capital A and a capital B. Okay, remember that outside represents possible gamete combinations. Then I do the outside, okay? So that parent could give a capital A and a lowercase b. So that's another gamete combination. Then I do my insides, okay? So that's gonna be little a, big B. I'm gonna keep them in that order. Remember, these are not genotypes, okay? And then I do the last of each letter. So little a, little b. And that is how we foil, okay? So you do the first of each letter, the outsides, the insides, and the last of each letter, okay? So if you're looking at it, the first letter goes to each of the second letter, and then the number 2 spot goes to each of that second letter, okay? So let's do one more practice. So letter B is lowercase a, and then capital B, lowercase b. So if we're gonna foil this one, first would be little a, big B, outer, little a, little b, inner, little a, big B, and last, little a, little b. And it's completely fine that they are the same, okay? You won't always get 4 different gamete combinations. If you had a parent that had all little A's and all little B's, then the only thing that they can give is little a, little b. So sometimes you don't necessarily have to foil, but just out of being consistent and trying to avoid mistakes, I always foil my parent genotypes. So this is the only thing that we're really adding, okay? Now, the next step would be to cross. Now, inside, I do want you to remember that the inside does represent genotypes. So like if you look at my combination here, once again, it's whatever's above and beside that's going in that box. I'm gonna put my A's first, capital A's first, and I'm gonna put my B's second, capital B's first, if I have them, okay? So like in this box here, that baby would get capital A, lowercase a, and then it would get capital B, capital B. So if those were my parents, that would be one possible baby, okay? And then I go through and I just fill out all of my 16 boxes, but you do want to make sure that you put your like letters together, okay? So my A's and my B's. All right, so that takes care of number 7. So now what we're going to do is we're going to do a sample problem, and we're going to take a look at number 8, okay? So this one says that this is the most complicated, okay? And what it means by that, it's not that it's any harder, but there's no shortcuts on this one. So if you look up in the information above, kind of in the middle, right after number 7, it says for any dihybrid cross, there are 9 possible genotypes, okay? And it's got them listed out there under where it says the 9 possible genotypes, all right? So the reason I say this one is the most complicated is this is the one problem that's going to give us all 9 possible genotypes, okay? There's also going to be 4 possible phenotypes. Now, if we're looking at 2 traits, then our phenotypes are going to include 2 traits. So it's like, it's no longer, is it purple or white? Now it's, what are the chances that my plants will be purple and tall? Or what are the chances that my seeds are going to be round and yellow? Okay, so we're just looking at two traits together. So your phenotypes are going to show two physical characteristics. And so there's 9 possible genotypes. There's going to be 4 possible phenotypes for any dihybrid cross. Okay, now you may not get all 9, you may not get all 4, but in general there's 9 possible genotypes and 4 possible phenotypes. Now on the phenotypes, I'm going to come back to this, but I want to explain what it's talking about on your notes. It says that you could have both the dominant traits, and a lot of times I just simplify that and say dom-dom. And then you could have the dominant of trait number 1 and the recessive of trait number 2, so I call that dom-rec. And then you can have the recessive trait of trait number 1 and the dominant trait of trait number 2, so that's rec-dom. And then you could have the 2 recessive traits, so rec-rec. So if you're trying to write out your phenotypic ratio, it's always dom-dom, dom-rec, rec-dom, rec-rec. Okay, so if you have trouble remembering what the 4 are, you can kind of do DD, DR, RD, RR, if that helps you out. But that's going to be the 4 possible combinations each time. Now, it's important that you understand this dihybrid because this is what your essay is. Okay, if you look in your study guide, at the bottom, towards the back, the bottom of one of the last pages, it has TC essay. And essentially what you're going to have to do is do a dihybrid from scratch. So you're going to make a key for the traits, and you can do any traits that you want. You can make some up, you can do a mythical creature, it doesn't matter. You have complete control over this dihybrid cross. You just have to show that you know how to do a dihybrid. So you'll make a key, you'll pick 2 traits and make a key. You're going to have to show what the parents were, so you'll write out the parent genotypes. Once again, your choice. You're going to set up your Punnett square, you're going to do the cross, and then you're going to write out the phenotypic ratio. Okay, so that's why you need to understand how to do it, because you're going to have to do one from scratch. You're going to be completely making up that dihybrid cross. So if you understand how to do them, that essay should be really, really simple. Okay, it's kind of hard to mess that one up unless you just don't know what you're doing. All right, so let's go ahead and take a look at number 8. So it says in peas, tall is dominant to dwarf, and purple flowers are dominant to white. So the very first thing I would do is I would make a key. Now, you are welcome to use any letter of the alphabet. Okay, I'm going to use T for my first trait. So tall was dominant to dwarf. Now, the second one, P's are not necessarily great letters to use because big P and little p can kind of look a lot alike. You could use the W for white as long as you can tell the difference. Once again, you can use any trait that you want. I'm going to use the letter F for flower. So capital F, the purple was dominant to white. And I try to make sure that my capital F and my lowercase f definitely look different. So just as long as you can tell them apart, you can use any letter that you like. All right, so we did step 1, we made a key. Step 2 is to identify our parent genotypes, okay? So you don't necessarily have to write this out, but you might want to because we're going to be foiling. So it says, what is the possible phenotypic ratio if 2 heterozygous plants are crossed? So remember that hetero means different. It says heterozygous plants for both traits. So what that means is both parents are heterozygous for both traits. So I have a big T, big F, little f. That's how we write our genotypes. You put the 2 together, and both parents were that. Once again, this particular trait is going to give us all 9 possible genotypes and all 4 possible phenotypes. Okay, so we just did step 2. We identified the parent genotypes. Step 3 was to separate. Alright, so there's my Punnett square. I need to figure out how to set up my boxes, and that's why I do the foiling. So I take the first of each letter, big T, big F. Now remember, both of my parents are the same, so I'm going to put the same thing going down the side, big T, big F. I do the outsides, big T, little f. I do the inside, little t, big F. And then I do the last of each letter. Alright, so there I've separated my gametes now, okay? You should always have 2 letters above and beside the boxes, 1 of each, okay? If not, you're doing something wrong. You're going to have babies that are missing genes, okay? So you should have 1 of each letter above. And remember, these are not genotypes, so I do not have to write the capital letter first, alright? Now, inside the boxes are my genotypes, and so that's where I want to put my T's together, capitals first, my F's together, capitals first, okay? Now, for time purposes, I'm not going to go through and fill out every single box, okay? But I think it's important that you do that, okay? You need to go through and fill out every box. This is one where there's no shortcuts, all right? So like, for instance, big T, big T, big F, big F. Big T, big T, big F, little f. Now, when you've done these things 1,000 times like I have, you start to see patterns. Like all of these 4 are going to have big T, big T. These 4 down here are all going to be little t, little t. And then these 2 groups are going to be big T, little t. Okay, and they have the same basic pattern. So if you do these over and over and over again, you're going to find the same patterns. So you go through, you fill them out. And then what you have to do is you just need to physically go through and count. Okay, so for the crossing, you just fill out the insides. Okay, the last part is to answer the question. And this particular question said, what is the possible phenotypic ratio? All right, so a lot of times it doesn't ask about genotypes. It might, but as always, what is the question asking you? So I want to show you how to write out the phenotypic ratio. Okay, so remember, you have 4 possibilities. You could have dom-dom, which are the 2 dominant traits. So one possible phenotype is tall and purple, okay? And we're going to put how many of those we get. All right, the second possibility is dom-rec. I can have the dominant of first, recessive of second. So my babies could be tall with white flowers, and I'm going to put how many of those. Technically, there'd be colons just like we had before, okay? Now this is already written out to the side for you. I'm just showing you where they got it from, because on your essay, you're gonna have to do this yourself. All right, the next possibility is rec, dom, the recessive of the first, dominant of the second. So they could be dwarf and purple. And then the last possibility is rec-rec, the 2 recessive traits. So dwarf, and that's where I really think the key comes in handy, okay? At this point, you just count, all right? Now, if you had all of this filled out and you counted your tall and purple, you're looking for boxes that have a big T and a big F, you're going to find that there are 9 that are tall and purple, okay? When you count how many big T's but only little F's, you're going to find 3 of those. Okay, for dwarf, you're looking for only little t's, but at least 1 big F. You're going to find 3 of those. And then all little letters, there's only going to be 1, and it's going to be right down here in this corner. Okay, so this right here is what we call our phenotypic ratio. This is a particular 9331. Now, I've mentioned several times that you guys need to be familiar with Mendelian ratios. So this is essentially my F2 ratio for a Mendelian cross, okay? If it's a monohybrid, I always get a 3-to-1 anytime I cross heterozygotes. So this is my dihybrid ratio anytime I cross heterozygotes. So I told you earlier this was kind of the most complicated because we get all 9 genotypes and all 4 phenotypes. Just FYI, anytime you cross 2 parents that are heterozygous for both traits, you always get a 9-3-3-1. And if you know the dom-dom, dom-rec, rec-dom, rec-rec, then you know what traits those numbers go with, okay? It's always 9 double dominant, 3 dominant recessive combinations, and 1 double recessive, all right? Now, I do just want to quickly show you that you can do a shortcut. All right, so we're not going to do another example problem, but I just want to show you that you don't always necessarily have to fill out every box, and this is completely acceptable. So let's say that I have my Punnett square, all right, and let's say parent number 1 was recessive, okay? So I would FOIL all right, but let's say my other parent was completely recessive. Okay, this is parent number 2. They have all little letters. So I have little a, little b, little a, little b, little a, little b, little a, little b. Now, you see how all of these are the same? If you have a problem like this, you can fill out one box. So big A, little a, big B, little b. And if this whole row is the same, I know that I'm going to have 4 of those. Okay? So there are shortcuts. You don't always necessarily have to fill out all 16 boxes. If your gametes are the same, then a lot of times you can do the shortcut of drawing arrows. It's just whatever you feel comfortable with, okay? When it comes to the one that you have to do for your essay, remember you get to choose your cross. So you can do whatever cross you'd like, all right? So that should take care of our dihybrids, okay? When you go to your homework problems, your dragon genetics, you have 2 dihybrid problems that you need to do. Once again, Ms. Petty is going to put the answers on the board, so you should be able to check it to make sure that you're doing it correctly, all right? So I think that is numbers 9 and 10, if I'm not mistaken. Yeah, you have numbers 9 and 10, okay? All right, so we have 2 more types of problems that I'm going to talk about, and these are under the other types of problems, okay? So if you turn in your practice to the very end of your packet, okay, the very end of your packet, and as I mentioned before, there's some notes earlier on the back of the front page. So this right here, we did dihybrids. We looked at 2 traits, okay, and you saw how we went from a 4-box Punnett square to a 16-box Punnett square. Well, we could technically go bigger. We could do a trihybrid, we could do a tetrahybrid, and we could keep going. We could look at more and more and more traits, but that's a whole lot of boxes, okay? If we go from a dihybrid to a trihybrid, we're going from 16 to a 64-box Punnett square. Now, I don't know about you, that's a lot of boxes to fill out. So the good news is there's a shortcut, okay? Now, the only catch is the traits have to be independently assorted, okay? If your traits are independently assorted, which means their inheritance is separate from each other, there is a shortcut that we can do, okay? And it's just based on statistics. So imagine that we were doing a coin flip. Okay, what are your chances of getting heads when you flip a coin? All right, I'm sure you're sitting there going 50% or 1/2. So what are the chances of me getting 3 heads in a row? All right, now how would we figure that out? Okay, in statistics, if the events are independent, so what I get on the first flip does not affect what I get on the second flip, and that doesn't affect what I get on the third flip. Each flip is an independent event, okay? So if you have independent inheritance, if each trait is inherited independently, then we can use this same statistical method, okay? So I have a 50-50 chance of getting heads, which for this particular method, always use fractions, okay? It's going to make your life much easier. So what are my chances of getting 3 heads in a row? Well, how I would calculate that is I have a 1 out of 2 chance on flip number 1. I have a 1 out of 2 chance on flip number 2. And I have a 1 out of 2 chance on flip number 3. So to figure that out statistically, we just simply multiply each of those probabilities. And so when you multiply fractions, you multiply across the top, and then you multiply across the bottom. Now be careful here, do not add. Okay, this is multiplication. So we have 2 times 2 is 4, 4 times 2 is 8. So you have a 1 out of 8 chance of getting 3 heads in a row. Okay, this is the same way that we're going to do some other types of genetics problems. So if you look at number 5, it says a parent with the genotype. All right, we have big A, little a, big B, little b, big C, little c. Now I don't love C's. I would definitely be careful when you're using C's. Make sure you can tell the difference. So it says that parent is crossed with a parent that is big A, little b, and big C, little c, okay? So we don't actually have to do a 64 box Punnett square. What we can do is we can do 3 little monohybrids, okay? So what I'm going to do is I'm going to do a cross for my A's because they're independently inherited. It tells us that. I'm going to do a cross for my A's, a cross for my B's, and a cross for my C's. And then I can just apply this statistics method, okay? So over to the side. I know this isn't going to be super big, but I'm going to do a big A, little a, cross with big A, big A. All right, for my B's, I have big B, little b, cross with little b, little b. And then for my C's, once again, be careful with these. I have big, little, and big, little. All right, so these are my independently inherited traits, okay? And now what I can do is I can just look at my probabilities of different outcomes. So the question says, what is the probability of having a child with 3 dominant traits, okay? So once again, use fractions here. If I look at my A box, all of them are going to be dominant, okay? So that's 4 out of 4, or that is just simply a 1. All right, remember I'm going to multiply my events together, so I have pretty much 100% chance of getting a dominant A, all right? When I look at my B's, 2 of them have capital B, so I have a 2 out of 4 chance. I would always reduce because it's easier to multiply. So I have a 1 out of 2 chance of getting a dominant B trait. And then for C's, I have big C, big C, big C, little c, big C, little c, little c, little c. So there I have a 3 out of 4 chance of having the dominant trait. Once again, the question asked, what are the chances of having a baby with all 3 dominant traits? So I just take the individual probabilities for each trait and I multiply them together. So I had 3 out of 4 chance of having a dominant for the 3rd trait. So once again, I multiply across the top and I get 3. I multiply across the bottom and I get 8. So I have a 3 out of 8 chance of getting a baby that is all dominant traits, okay? Now, what that means is, if I had 64 boxes in my trihybrid Punnett square, I would get the ratio that would equal 3 out of 8, okay? So 24 out of those 64 boxes would be all dominant, okay? So this is actually going to be a lot faster. Now, when you go to do your practice problems on this, let's say I was crossing a parent with itself, because one of the examples says they self-pollinate, okay? So what that means is you're crossing that parent with itself. So like you see how if I'm crossing a big A little a and a big A little a, and a big B little b and a big B little b, I don't necessarily have to do 3 separate boxes. If it's the same cross, you don't have to do it 3 times, okay? You're going to get the same ratio whether it's A, B, C, all the way through Z. So you just need to do the boxes if they are different crosses. All right, if it's the same cross 3 times, you don't have to do it 3 times. You just need to know what the ratios are going to be, all right? So if you get more than a dihybrid and they are independently inherited, all you have to do is just multiply your individual probabilities for each trait, and your answer should come out in fraction form, okay? So when you go to your Dragon Genetics packet, there is a section that is called Other Problems, okay? It's numbers 13, 14, and 15, okay? Numbers 13 and 14 are just like what we did here, okay? You're going to do some monohybrid crosses, and then you just multiply the individual probabilities, okay? You do need to show your work and not just put an answer out to the side, but your answers need to be in fraction form, and they need to be reduced, okay? Reduce them to their lowest level. So if you have any questions about that, get help, feel free to email me, or feel free to talk to Miss Petty. But this right here is an example of how to do that, and you can always come back and look at this one, all right? But once again, you'll also have the answer key to check to make sure you know what you're doing. So that's how you're gonna do number 13 and number 14, okay? Now, the last ones we're gonna talk about are the problems that are like number 15. And these are gonna actually require you to work backwards, okay? These are the only ones that we're not necessarily gonna follow those 5 steps, okay? Because what it's doing is it's telling you, these are my babies, what could the parents be, okay? So if you go back to your note packet, you do have a sample problem like this. It is at the very back, it is the very last problem, it's number 6, okay? And so on number 6, very back of your practice packet, it says purple flowers are dominant to white and tall is dominant to dwarf, all right? So just like we did before, I would make a key. It's gonna make your life a little bit easier. All right, so I'm gonna use the same letters that I used earlier. So purple is dominant to white and tall is dominant to dwarf. So you can make yourself a key and use whatever letters you want to. All right, now what they're giving us, so this is a dihybrid cross, okay? What they're giving us are the babies. They're saying this is how many of each baby that I got, okay? So if you look at your numbers on A, we had 95 that were purple and tall. There were 29 that were purple and dwarf. There were 33 that were white and tall, and there were 11 that were white and dwarf, okay? Now, this is what I would recommend, okay? Instead of looking at it like a dihybrid, I would look at it like 2 monohybrids, okay? Now, if you know your genetics ratios, and I see 90, 30, 30, 10, that's a 9-3-3-1 ratio. So if you're already familiar with your ratios, we should know what this outcome is going to be. But I want to show you how you get it if you don't know, okay? So what I'm going to do is I'm going to look at it as trait number 1, and I want to kind of get a general total, all right? So I want to know how many purple babies I had. So I had about 95 there. I had 29 there. I'm just going to round that off to 30, okay? So I had about 120 purple babies. Once again, I'm only looking at trait number 1. And I had about 40. Doesn't have to be exact. All right, so I had a 120 to 40 on purple and white. Now I'm going to do my tall versus dwarf. I had 95 and 33. Once again, that's about 120, 125. And I'm going to look at my dwarf. I had 29 and 11. Okay, once again, about 40. So if we look at these as individual monohybrids, okay, what I have is about a 3-to-1 ratio, okay? So a 3-to-1, a 3-to-1, and this is where it just tests your genetics knowledge, okay? If you don't know where that comes from, you may want to do some practice crosses. I hope that you know that the way you get a 3-to-1 is any time that you cross a heterozygote with a heterozygote, okay? So just to kind of show you how do I get a 3-to-1 ratio, it's when you have 2 heterozygotes, all right? So my answer here is what could my parents be? What parents produced these babies, okay? So this was my given. I'm working backwards. So the only way that I could have gotten those numbers is if, for the trait number 1, the parents had to be heterozygous. And then for trait number 2, once again, I got that 3-to-1 ratio. So they must have been heterozygote for trait number 2 as well. Okay, so this is my answer to letter A on number 6 on your practice packet. Okay, so if we look at that, we can see that 9331, and as soon as you see that, you know it's heterozygote for both traits, or I can work it out as little monohybrids. Okay, now let's look at one more example. Let's look at letter B. Alright, so we still have the same key. Okay, I'm not going to rewrite it for time's sake, but we still have the same key. But this time we have 74 white and tall, and we have 28 white and dwarf. So obviously this one looks a little different, but once again, look at it in terms of 2 separate traits. Okay, so trait number 1, purple versus white. If you notice, all of my babies are white. Okay, all of my babies are white. And if we go back to our key, white was recessive. Okay, so using our genetics knowledge, the only way that all of my babies can be recessive is if both of my parents are recessive. Okay, because if either one of those parents had a capital F, 100% of their babies would not have been recessive. Okay, now when we look at trait number 2, I now have 74 tall and 28 dwarf. That is about a 75-25. That's going to give us about a 3-to-1 ratio. And hopefully by now we know that a 3-to-1 is always going to be the end result of 2 heterozygotes. So for trait number 1, the parents were all recessive. For trait number 2, my parents must have been heterozygous because I have a 3-to-1 ratio when it comes to tall and dwarf. So both parents are heterozygous. So these are my parents that could have produced those babies. All right. Now these are typically some of the hardest for students because it's making you work backwards. It's really testing your knowledge of genetic ratios. When in doubt, if you don't know how you get a 3-to-1 or you don't know how you get a 100%, you can do test crosses until you figure it out. Okay. But this time we're looking for the parents that produce these offspring. Okay, so make a key, look at them individually, and then you can figure out what your parents could be. Okay, and so in your dragon packet, this is going to be number 15. You have 4 different examples, all using the same trait. The answers are going to be on the board, and if you have any questions, just get help. Okay, so on your next quiz, you're going to have a set of notes that we're going to be covering, and you're going to have the dihybrids, other problems. We're also going to add blood and sex linked. Okay, so just kind of be prepared for that. All right, hope you guys have a great day. Please refer to the video or look up other videos on YouTube if you have any questions about the problems we covered today. All right, y'all have a good one.